Thank you. Kia ora koutou, ko James McCarthy aho. I'm a plant ecologist at Manaaki Whenua Landcare Research, which is one of the New Zealand Crown Research Institutes. So the work I'm presenting today was published in the Journal of Applied Ecology all the way back in 2021, and it concerns the concept of refugia, which is areas of sort of safe haven that support populations of a species that was once more widespread. People usually talk about this in relation to climate change studies, where species manage to hang on in places like mountaintops during glaciation events, from which they can spread again once the climate warms. So here we apply the refugia concept to myrtle rust, where we quantify how much refugia New Zealand's Myrtaceae species may have. So how much of a species range might occur outside the disease range, mapping where that might be, and also how you might be able to restore forests to provide additional refugia. So the first step was to produce some maps predicting the distributions of New Zealand's native Myrtaceae using species distribution models. Now, when you think about a species like Pahutakawa or Metrosideros excelsa, which we have in the pictures here, you think of a species that usually grows on the coasts in wetter, warmer, sort of northern areas. And this is the kind of information or data that we use to predict a species distribution based on the characteristics of the environment from which we know that it occurs. So using vegetation plot data, we know that Pahutakawa is found in these locations. So as I mentioned before, warmer, northern, sort of coastal areas. And it's absent from these locations. And it's these presence and also crucially absence locations that we use to build models which produce relationships with a bunch of environmental variables as predictors. And these models can then be used to predict a species spatial distribution to fill the gaps between the point records for where we know a species occurs or doesn't occur. So here's an example of the prediction for Pahutakawa. Pahutakawa is actually a bit of a special case because planted individuals of this species occur as far south as Christchurch, and it's also widely planted on the Wellington waterfront. But for this exercise, we're just interested in the natural range of Myrtaceae, so excluding planted individuals. And as you can see, it fits pretty well with the point records that I showed you before, which are in red. And it also produces a distribution that predicts a little further down both coastlines, which is about right for what we know for where the species occurs. Sorry, there's the points there. So distributions were produced for almost all of the native Myrtaceae. There are a few missing. We didn't have enough distributional data to produce models for Bartlett's rata. We actually know where every single one of the individual of those species is, probably anyway, so it wouldn't have been quite so useful. Same for Metrosideros kermiticensis, which occurs on the Kermadec Islands, north of New Zealand, and one of the climbing rata vines, Metrosideros carminia. And we also lumped the 10 species of Carnuka, which is Kunzia ericoides at the top left. And since then, there's been a proposal to revise that taxonomy anyway. So there's a bit of flux. The status of that species is in a bit of flux. And most of the distributions that we produced make sense. So the two at the top left, they're Carnuka and Manuka, respectively, from left to right, really widespread species, successional species that occur in landscapes that were once cleared normally. Metrosideros umbellata, which is sort of in the middle on the right there, southern rata, you can see it occurs quite far south. So generally they weren't too bad. And in the paper, we present results under two myrtle rust scenarios. And the justification for the myrtle rust distribution that we chose is a little bit complicated. And the reality is likely probably to be somewhere between the two, because we do know that it occurs quite commonly around Wellington, also in Marlborough, and it's also actually spread to Christchurch since we did this work. So the results I'm presenting today will be from scenario one, which is probably the more optimistic of the two. But it probably captures a more core range of the disease where the effects are likely to be more severe. And these maps are based on the work by that Rob Beresford and colleagues published in 2018. That's scenario one, the one we're presenting on. So here's a distribution for Rama, the species we've been talking about quite a lot. Lophomertus ballata, probably the most severely affected species that we have here in New Zealand. Sorry if it's a bit difficult to see, but there should hopefully be some red areas, some red sort of blotchy spots, which is kind of typical for what we know about Rama. Actually, it tends to be quite common and abundant when you do find it, but it's patchy. It's sort of here and there. We calculated that this species has a range of about 614 kilometres squared, of which only 15 kilometres squared, or 2.5%, occurs outside the disease range for the scenario that I'm presenting on at the moment, which is the map with the yellow to the left. So not very much, and even under that optimistic scenario that I was talking about. Looking on the positive side, there are probably about 120 kilometres squared of areas that are climatically suitable to the species that you could restore to provide additional refugia. So these are areas that are currently under a different land use at the moment that doesn't support forests. So it could be some sort of, most likely agricultural land that could be restored. And just to show another example for another species, southern rata, Metrosideros umbellata. I didn't actually know that this species had been infected until I looked up the list again the other day, and Rowan mentioned it yesterday. There have been infections on this species, but not many, and I think Rowan said mostly from nursery individuals. So this is a species that has a really southern distribution, as you can tell by the name, although it does actually have little populations up in the very far north. And this is an example where most of its range, 90%, is outside the disease range that we're talking about. So this might be a species that would be of perhaps of lesser concern, even if it was infected, at least immediately. It might be of lesser concern. And there's also a small amount of restorable refugia for this species. I'll just take a quick moment to say that all of this work isn't intended to sort of diminish the importance of plants in place. That's, of course, crucial that we keep populations of these plants within their ranges, close to where they should be. But I think this is another sort of useful tool that we can use as part of the response. And just finally, this table here shows the New Zealand Myrtaceae that we assessed ranked by their refugia. So you probably only really need to look at that dark grey column, which shows the percentage of refugia that they have. So as you can see, the situation for some of these species is pretty dire. For example, the top one, Metrosideros albiflora, which is in one of the climbing rata or arca vines that Hone was talking about before, has only 0.1 of its percent of its range outside even the most optimistic scenario. This is a really northern species. So this is a climbing rata. I don't think many people know what it looks like, let alone climb to the top of 35-metre trees to check. So this is probably one that, if it was infected, it may not be detected. And even if it was detected, it would likely get infected throughout its entire range. Syzygium maire, or Maire tawake, is the next one down with 0.6 of its range. So it's in a similar boat, and this species is quite severely affected. So these are examples of species that probably can't rely on refugia for their conservation and protection, so we'll need to come up with other methods to safeguard them. Just some ideas of where we might go next. I mean, of course, improved data means more accurate models, so more information about the distribution of these species would enable us to produce better distribution maps for them, especially for those that aren't particularly common, like Syzygium maire. Incorporating refugia into the response, so sort of species prioritisation. I think this has a role for that. You could consider translocation for extremely susceptible species, and the range maps could be used to help target seed collection and also sampling for genetic diversity and resistance screening. I've got some acknowledgements, but that's it from me. Thank you.
Thanks, James. If you cast some of the climate change scenarios across your refugee areas, does it get better or worse in terms of amount of land or space that might be available?
Yeah, so there are sort of two components to that, I suppose. One is the change in the distribution of the disease, which is probably the one you're talking about. And I gather it would probably get worse. I mean, myrtle rust likes warm and humid kind of areas, so those kind of environments are just going to move further south and cover more of the country. Rob might be able to give me a quick sort of nod or a shake of the head if the top would become less susceptible. I don't think that would be the case. It wouldn't shift down with the top becoming free, would it? Climate change is bad news, thank you. And the other side of it, I mean, so something with arms and legs and wings and flippers can respond quite quickly to climate change, but plants can't. A rust can probably move about and respond quite quickly to climate change, but the plant communities aren't going to, and they're going to be stressed for a whole bunch of reasons. So with rapid climate change, you're not really going to see plants respond in the way that they would under a more normal kind of regime, so it's probably not going to be looking good. And it would be interesting to sort of formally analyse that, but we haven't yet.
I have a question, a little bit related to that, I suppose. The refugia that you have identified now, do you know for certain whether these are areas that the disease can't go, or is it currently just probability that the disease just happens to not be there yet? And I guess what I'm wondering is that if you use refugia, how do you know that the disease won't just spread into these refuge areas because it has just been probability that it wasn't there yet, rather than there is a distinct reason for the disease not to survive in those areas?
Yeah, it's all based on probability. The maps for the disease range that we worked on, there's a probability of infection risk basically everywhere in the country. So it's almost a conservation decision. What kind of level of risk are you willing to accept? It might be that you're willing to call it refugia if there's only a small amount of infection. You know, the conditions aren't quite right, but plants will survive. But I think that's a tricky. I think it's a conservation decision. That's where I arrive in my head. What kind of risk are you willing to accept?